Assalamualaikum and a very good morning to Madam Dia, our lecturer for code CHM260 Basic Instrumental Analysis. I am Amir Ilham bin Muhammad Saleh from class RAS1154C.
Hi, Assalamualaikum. My name is Fatir Maira binti Muhammad Nordin.
Hi, my name is Afika binti Abu Bakar.
Okay. Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Izzat bin Ahmad Asim.
The first question that we should ask when we are talking about this topic, why we analyze lead in water, is how does lead came into contact with water in the first place? The general source of lead that is being found in water is the components of plumbing that is being used that is made out of lead. The corroded surface on those components is the main source why there is a trace of lead in water. These types of plumbing components is usually found in older generations' homes, since in those days lead is being used as the plumbing components, which has the characteristics of resisting pinhole leaks, and it is easy to shape into a component that can deliver water. But in the new generation, when there is an advancement in chemistry, they found out that lead can cause a lot of health problems to humans. Thus, the invention of PVP pipes and PEX pipes is being used to substitute those components in the new housing plumbing. The next source that contributes to the amount of lead in bodies of water is industrial waste. Industrial waste that came from industries such as manufacturers, mining, contributes to the amount of heavy metals in water, which include lead. Yes, these industries does provide jobs, services, and products to the community. But are we ready to face the devastating effects of the amount of heavy metals releases into the body of waters, which include lead? It will not only affect us humans, but also other organisms and the environment. Other than that, the unexpected source of lead that can came into contact with water is the improper disposal of electronics devices such as computers, TV, since these devices contains components that is being made out of lead. When they get corroded, if a rain came by, the lead will get washed away and end up in a drain, which will eventually get into larger bodies of water. Moving on to the next point, we are introduced to BLL. BLL, also known as blood lead level, is the method that is being used to analyze the amount of lead in a patient's body. The normal amount of lead in human's body is being classified into three different groups. The first group is adults, the second is children, and the third is pregnant women. For adults, the normal amount of lead in their body is supposed to be 10 microgram per deciliter. For children and pregnant women, the preferable amount of lead in their body is 5 micrograms per deciliter. The first effect is cardiovascular diseases, which is hypertension. The first effect is cardiovascular diseases, which is hypertension. which can be explained by the nephrotoxic actions of this metal towards humans' bodies. The hypertension effects that is inhibited by lead in human body are impact on cardiac structure, effects on structure and function of endothelium, mechanism of reflexive blood pressure, enhance free radical generation, and increase plasma renin activities. Next, there is evidence that as the concentration of lead increases in an individual's body, the glomerular filtration rate decreases. Glomerular filtration rate is the rate of the functions of the kidney. If the rate is high, your function is good. If the rate is low, your function have deteriorated over the years. A person with a low GFR tend to have a higher risk of getting a chronic kidney disease, or also known as CDK. Lastly, lead too affects the fertility rate in adults. Amongst the effects are disturbing the reproductive hormones, disrupting ovarian functions, the egg quality, overall fertility and pregnancy outcomes. The effects of lead on fertility affects men as it can disrupt the regulation of spermatogenesis and the sperm development. Ooh, that is not good. Moving on to children, a high level lead exposed towards them can cause a permanent brain and central nervous system disruptions, which could cause behavioral problems and mental retardation. In some cases, the IQ will be reduced. Lead can cause hearing problem as it is related to degeneration of cochlea, organ of Corti, ganglion cell loss, strial atrophy, and lastly, basilar membrane stiffness. In some cases, a high level of lead in children's blood can cause anemia. When the lead level in children's blood is more than 10 micrograms per deciliter, will cause a decrease of iron absorption and hematological parameter affection. The last group is pregnant women. The effects of lead on pregnant women is that the fetus that they carry will have a significant reduce of weight. This is due to lead competing with calcium to be deposited into the bones, which will causes the fetal growth to be forever impaired. Also binds with sulfhydryl groups and inhibits enzyme involved in heme synthesis, which is important for cellular respiration and metabolism, as well for hemoglobin synthesis. This will contribute to the reduce of growth of the fetus. Other than that, lead can cause premature birth, as lead can lower the concentration of E2 serums in pregnant mothers. Lower E2 levels have been associated with increases of risk of spontaneous preterm birth in epidemiology studies.
I'm going to talk about the techniques we can use in this experiment. To determine the leads at trace levels in water, we have 3 methods to proceed with this experiment. So, the first one is electrothermal atomic absorption spectroscopy, and the second one is flame atomic absorption spectroscopy, and the last one is the anodic stripping voltammetry with microelectrode. So, the first technique is electrothermal atomic absorption spectroscopy. So, this spectroscopy is known as graphite furnace spectroscopy. So, here are the advantages of using this spectroscopy. Firstly, this spectroscopy, this system can detect small sample, and also the sensitivity for this detection system is very excellent. So, for this, the disadvantages of this system is it is a slow measurement time and limited analytical range, and also the maintenance is high and cost associated. Okay, next is flame atomic absorption spectroscopy. This one is a common technique we can use to detect metals in sample, and this spectroscopy is based on the principle that ground state metal absorb light at specific wavelength. So, ions in solutions are converted to atomic state by means of flame. So, when the light of correct wavelength is supplied, the amount of light absorbed can be measured, and a reading for concentration can be obtained. So, advantages of using this spectroscopy is we can find this in our lab, so it's easier for us to do this experiment, and the detection system is more simple and also cheap. When we use this spectroscopy, we can have a good quality, and also we can have a accurate quantity. Okay, for disadvantages is we can only analyze a solution, and also large sample is needed. Large sample quantities is required for this spectroscopy, and this spectroscopy is less sensitivity and have a problem with refractory elements. Okay, so the last method is anodic stripping voltammetry with microelectrodes. So the application of microelectrodes for microanalysis is demonstrated using a novel capillary flow injection system. So, heavy metals at micro 1 concentration have been determined in micro detail in sample. So, the influence of electrode size and convention during the depositions period of anodic stripping voltammetry on the reproducibility of lead at trace levels in water. So, that's all the method we can use to determine lead at trace levels in water.
I'm ready to present to you about our instrument AAS, or we call it atomic absorption in spectroscopy. Okay, today I want to explain to experimentation using atomic absorption spectroscopy. So in this experiment, we have to do an experiment. The article they have to do to determine the heavy metal ion in water sample by using the AS. So in this experiment, the use of chemical modified silica, yes, SiG, CHBPA, ion exchanger for removal, and pre-concentration of chromium, manganese, ferrum, cobalt, nickel, cuprum, zinc, cadmium, and plumbum in natural water sample collected. And also in the experiment, we have to determine the effect of percentage of recovered metal ion, including mass change of ion exchanger, stirring time, pH of sample solution, and eluent concentration were studied. So after we have to know the experiment, what we have to do the experiment, we have to go to the experiment. In the experiment, we have to use the chemical modified silica gel, CIG, CHBPA, or and gas 1 carboxy 6 hydroxy benzylidinine propylamine. So the next is we are using to five apparatus: atomic absorption spectrometry (AAS), infrared spectra, electronic spectra, pH saturation and thermal analysis (TG). And next we have to know the synthesis of modified silicon gel phase, because this gel we have to modify. Yeah, so there are many instruction about modified. This is synthesis of the silica gel. Okay. Next we have after we have do the experiment, we to know the result and discussion. So, and in the experiment, there you conclude the result of discussion completely in bind with each other. So, in this is key point of our discussion and result: the investigation for uptake behavior of SHGHPA, ion exchanger effect of pH, effect of mass change of ion exchanger, time of stirring, concentration of eluent, acid, EDTA, and some interfering species, separate separation of chromium 3 and manganese to obtain from the chromium and permanganate, nature of bonding between metal ions and SiGHIPA, ion exchange infrared absorption and reflectance of and UV spectrometry. Also, probably mode of chelation of SiGHPA, HBPA or ion exchanger with metal ion, potentiometric measurements, TG and DTG analysis of solid unloaded offloaded CHG, HBA ion exchanger, comparison between log K values of loaded metal ion and active silica gel alone and modified inside GCHBA, and lastly the water analysis. So in this article, they are very long that they want to explain the result and discussion, and this is their discussion and result for the experiment. Okay, this is the result and discussion for this experiment. So next, after we have to know why this experiment use the AAS, so the importance of using why atomic absorption spectroscopy AAS important in this experiment, because atomic absorption spectroscopy is to analyze the concentration of element in a liquid sample based on energy absorbed from the curtain wavelength of light, and also they widely used in inorganic chemistry to determine the total element concentration in many sample types and generally allow rapid sample throughput. Okay, this is only for our discussion, so we can thank you very much for hearing this.
Next, we'll move to question 4. The question is how the cited experimental data can be used to achieve the objective of the study. So first of all, what we have to know is the objective of this study is to determine the amount of fluid in water body. As you can see from table 4, the concentration of the heavy metals ion is increases from cadmium to cuprum to the plumbum, clearly in a region starting from Al-Manzurachiti till reaching its highest value at diameter bridge, and the highest concentration of lead is 10.2 micrograms per liter. In addition, EPA set maximum allowable concentration of lead, which is 15 micrograms per liter. So this is enough to prove that the study is success. The advantages of using these articles compared to others is this article or this study is using two methods to get the better or the exact result. What two method uses is the first one is ion exchanger and the second one is solvent extraction. So first of all, I will explain what is ion exchanger. Ion exchanger are sphere of sulfonated synthetic resin that can take ion from liquid, water, condensated water, processed water, swimming pool water, by changing them out against others ion. The liquid will flow over a width bed filled with ion exchanger resin column. As we know, everything has a pros and cons. It seems with these ion exchangers, which it have advantages and disadvantages of using this method. So I will tell the advantages first. So the first one is its selective removal is possible by choosing the right ion exchanger. The second one is high removal efficiency is possible for cation and anion. For the second one, regeneration of the bed gives a very long service life. And for one, ion exchangers can also work well with low pressure. For the disadvantages is disposable cost of the acid and base for regeneration. Resin can be sensitive for oxidizing substance, and the third one is sensitive to pollution, thing, for example, of suspended of particles. And the second one, we are going to solvent extraction. So what is solvent extraction? Solvent extraction is a process which compounds are separated based on their relative solubilities. These treatment methods involve using a solvent, which is a fluid that has the ability to dissolve another substance. The advantages using this method is short extraction time, which is it's simple instrumentation, reduce solvent use, short extraction period, and it's also environmental friendly and high speed of analysis. But the disadvantages is its high capital cost, requirement of high pressure, and it have chances of impurities. So what I can conclude from this is these two methods have no significant difference was found between the values obtained by method 1 and method 2. So study this study is strongly accepted. Thank you. Last but not least, I want to thanks to Madam Ya for all the dedication of teaching CHM 260. Without you, we all can't reach this part.